Friends, welcome to Biology Made Easy. Today in this video, we are going to discuss the Hardy-Weinberg principle. So there is one English mathematician known as G.H. Hardy, and one German physician W. Weinberg. Both of them in 1908 independently produced certain views, certain principles to correlate how the evolution is taking place in the population. And this is known as Hardy-Weinberg principle or Hardy-Weinberg law or Hardy-Weinberg equilibrium. Now this theory given by Hardy and Weinberg, it is only a theoretical concept. It is only, we can tell that it describes only theoretical situation under which evolution is taking place. So this is important. It is only a theoretical situation. So what is the definition of this law? What is the definition? Now according to this Hardy-Weinberg principle or law, all the frequencies of alleles in a population is always stable and constant generation after generation. I am just writing it. That is, the law states that the allelic frequencies in a population is stable, important, and constant, important from generation to generation. So according to this law, the allelic frequencies in a population, this is also important, not in individual. So the allelic frequencies in a population is always stable and constant, generation after generation. That means generation after generation, the allelic frequency never changes. But one condition is there. What is that condition? Here, as long as, that means here there are certain assumptions. And if those assumptions are accepted, then this holds good. So what are the assumptions? Now the assumptions are, number one is population should be very large. Then this is possible only when the population is in large in number, not less. The second point is there is a random mating between individuals, random mating. And the third point is there should be no mutation. And the fourth point is all the offsprings must survive and should have equal reproductive capacity. That means Hardy and Weinberg law or principle holds good as long as these things are assumed. That means the population should be very large. There should be a random mating between the individuals. There should not be any mutation taking place, and all the offsprings that will be formed in generation after generation, all of them must survive, and they should have equal reproductive capacity. Then only this statement holds good. Now, what is the explanation for this? Now, let us discuss the explanation. How they explain it. Explanation. So, according to Hardy and Weinberg, there is always in the nature, there are different allelic frequencies, there are different variations, but in spite of the large number of variations in the genotypes, why the evolution is not taking place, or how the evolution is taking place. So that is, let us explain it, that how the, in spite of the large variations in the genotype, the evolution or the variations remain constant generation after generation, and you can tell that it remains unchanged. So let us give the explanation. Now suppose let us assume that in a sexually reproducing diploid organism, Now here this diploid is important, also sexual reproducing is important. Why we have taken the diploid? Because there will be two alleles. So suppose in a sexual reproducing diploid organism, there are two alleles. One is capital A, which is dominant, and other is small a, which is recessive. And again you suppose that the gene A has frequency of allele Q, and A has frequency of alleles, this is P, and A has allelic frequency Q. And in nature, always P plus Q is equal to 1. So, if in a diploid organism, the dominant allele is A and the recessive allele is small a, and the frequencies are like this, then what is happening? Let us see. Now let us write here. Suppose this is a allele and this is the frequency. Then allele A, it has frequency P. Then what will be AA? Naturally, it will be P square. Then the recessive A, its allelic frequency is Q. Then what will be AA? It will be Q square. Then what will be when it will be PQ? When it will be AA, then the allelic frequency will be PQ. But what actually happens when a cross is made between two? Suppose one is AA, the other is small a. So the hybrid is AA. Now when this is intercrossed, now again we get AA on the male side and AA on the female side. Now if you draw the checkerboard, what we'll get? We'll get the dominant homozygous one, then this is the heterozygote, this is the heterozygote, and this is the homozygous recessive. That means we are getting the dominant homozygous in one proportion, then the heterozygotes in two proportions, and the homozygous recessive in one proportion. Or we can tell that this occurs in 25%, this occurs in 50%, and this occurs in 25%. So that means if the heterozygotes are 2 in number, here we can write 2AA will be 2PQ. That means now if we will apply the binomial expression, then what we will get? We will get P plus Q whole square is equal to P square plus 2PQ plus Q square is equal to 1. Because PQ is always 1 in the population. Now if this is correct, then what is this P square? P square is the frequency of alleles of homozygous dominant, and Q square is frequency of homozygous recessive, then 2PQ is the frequency of heterozygotes. So from this we can mark one thing. That means the gene or allelic frequency, that means the allelic frequency, we can write P square plus 2PQ plus Q square. And what is the genotype frequency? Allelic frequency of alleles. Then the frequency of genotype, it will be AA, 2AA, and small a. Now from this relationship, from these two equations, one is the frequency of alleles and the other is the frequency of the genotype. From these two, we can conclude that a relationship exists between the frequency of alleles and the frequency of the genotype. That means if we can know the allelic frequency, we can calculate the frequency of homozygous dominant, homozygous recessive, and the heterozygotes. And just in the reverse way, if we know the frequency of homozygotes or heterozygotes, we can calculate the frequency of alleles. Then you may now ask, how? So how can we know? So let us give some examples so that your understanding will be clear. So by giving examples, it will be very much clear. So that any problem comes in the examination, within 10 to 15 seconds, you will be able to answer the questions. Now, how can we know? By knowing the frequency of alleles, the percentage of homo and heterozygotes, or the vice versa. Let us take some examples. Suppose, let us take example number 1. And the example number 1, suppose in a population, the frequency of P is 0.7 and that of Q is equal to 0.3. Then calculate the percentage of homozygotes and heterozygotes. Now you see, here the P and Q are given. That means P is equal to 0.7 and Q is equal to 0.3. Now, what will be P square? So 0.7 square means it will come to 0.49, and what will be the Q square? It will come to 0.09, and what will be 2PQ? Now 2PQ will be 2 into 0.7 into 0.3. So it will come to 0.42. Now if we will apply in the formula, then P square plus 2PQ plus Q square is equal to 1. Now this P square is 0.49, 2PQ 0.42, Q square is equal to 0.9. You calculate how much it is coming. It is coming to 1. So if this is the equation. Now how will you know the percentage of homozygotes? Now you see, AA plus 2AA and the small a, this will give you 100%. Now what is the frequency of this? Now the frequency of this is 0.09. That means the percentage of homozygotes is 49%. This is 42% and this is 9%. So 49% means these are homozygous dominants. These are heterozygotes, and these are homozygous recessives. So again you calculate 49% as 42% and 9%, it is also coming to 100%. So in the question, calculate the percentage of homozygotes and heterozygotes. So the dominant homozygotes are 49%, the recessive homozygotes are in 9%, and the heterozygotes are 42%. That is when one is given, the other we are calculating. Now let us take the reverse one so that it will be still more clear to you.